Now we have the last talk. Dr. Lloyd Jeffs, Senior Director of BioPharma Services at Precision NanoSystems from Vancouver, is talking about the development of effective and safe RNA-LNP medicines for the clinic.
Can everyone hear me? Very good. First of all, it's a privilege to be here, and I want to thank the organizers. This is a great event. Already heard some great presentations in this session and the early sessions today and yesterday. I've been very grateful, I am very grateful for being working with liposomes and lipid nanoparticles for the last 25 years in the industry. And I've worked for 3 companies, and all 3 companies have been founded by Dr. Peter Cullis, biochemistry professor at University of British Columbia. So in a way, my career is indebted to Peter, because without the companies he started, I don't know where I'd be. So I'm here because of him today. So I'm going to talk a little bit about the development of effective and safe RNA-LNP medicines for the clinic. This is the work that we've been doing at Precision NanoSystems. We are a global leader in nanoparticle solutions. As you can see that we have produced and placed over 900 of our nanosembler particle formation systems over the last 10 years. We also have a BioPharma services group that I lead, and we've completed almost close to 200 projects for clients. We do not develop drugs, we support other companies in the development of their nanomedicines. Okay, so that's an important distinction. We have a new facility. I don't know if the pointer works on here, but you can see in the bottom left-hand corner, we are building a new facility in Vancouver, Canada. That's where we are based. Our headquarters are. And we'll have GMP manufacturing capabilities coming online in 2025. So if you're in the Vancouver area toward the end of 2024, beginning of 2025, we'd love to show you around our labs and our new GMP clean rooms. So we offer a full stack of technology to enable genomic medicine. And it's a revolution, it truly is. So in the next 5 years, I'm really excited to see the drugs that are going to move through development to approval. And we developed a genomic medicine toolkit. Initially, we started with our nanosembler manufacturing platform that is in the right side of the box. But we've also, basically due to needs and requests from clients, developed a Genvoid delivery platform, which are reagents and kits and proprietary ionizable lipids. And we have design capabilities around plasmid DNA that allows for the production of messenger RNA and self-amplifying RNA. So if a client comes to us, they can come with their disease target. We can design the pDNA, make mRNA through IVT at P&I or with our basically collaborators and sister companies. That's only possible because we have a large drug development team. And the expertise that we have spans engineering, process development, lipid chemistry, and we have probably about 150 scientists working at P&I right now who are completely focused on nanomedicines, specifically lipid nanoparticles, but also experience in polymeric nanoparticles and liposomes as well. So there's been some talks about extrahepatic delivery, but delivering mRNA-LNP to sites other than the liver, I think, is the current challenge. And it's great to hear some of the presentations from earlier today where there's a focus on doing that. And really, we can unlock the full potential by developing these extrahepatic solutions. So I'm really interested to see any new developments in that area. And P&I, we're eager to help, whether it's in cancer, rare diseases, or in cell therapy. So there's a number of versatile solutions that we have. So we can basically help clients develop vaccines, gene therapy solutions, or cell therapy applications. We also, we have a nucleic acids piece where I think, as many of you know, you can actually encapsulate many kinds of nucleic acids into lipid nanoparticles, and we have experience with basically all of them. And also drug carriers. And I'm excited because I think we've seen a lot of development and focus in the lipid nanoparticle space. Before the pandemic, we were seeing more and more projects in polymeric nanoparticles, hybrid-type particles, emulsions, exosomes, and traditional liposomes as well. And they're coming back. I really believe that. I don't think lipid nanoparticles are the solution for absolutely everything. I really believe in nanomedicines being the solution for everything, but there's many different types and flavors. So this is the projects, I think, at the end of 2022 that we've been working on, service projects. So for delivery route, it's an even mix probably between intramuscular delivery, vaccines primarily, and intravenous application. We've done a few projects with intratumoral, intravitreal, intratracheal. For payloads, it's basically mRNA and even split between mRNA and self-amplifying RNA, but other nucleic acid payloads as well, such as short interfering RNA, circular RNA, DNA, small molecules, and peptides. An application area, again, majority now is, and it's growing in the gene therapy space. I think there's still lots of interest in vaccines, but with the kind of post-pandemic scenario, the gene therapy is taking over again, at least with the projects that we've been working on. So P&I was formed basically out of a need to reproducibly produce nanoparticles, lipid nanoparticles, liposomes, in a way that you didn't have to have a PhD in liposomology, where we could provide a commercial solution at different scales. From the smallest scale, we can make a few microliters, all the way up to hundreds and hundreds of liters, using our next-gen technology. And it's based on Dean Vortex bifurcating mixers. And that allows for the very controlled and reproducible mixing conditions that are required basically to have consistent particle properties from batch to batch and when you scale. So it reduces basically the uncertainty, increases the reliability of the batches you're making, and those batches will go into experiments, and that will allow you to develop. So it helps, if you can make nanoparticles reproducibly, that de-risks your drug development programs, and it helps you accelerate those programs as well. So we provide commercial solutions for particle formation. We also provide reagents and ionizable lipids that you can use to help speed up your development. So scale up and scale down is very important. And this is our new system. We have launched it this year. It'll be available early next year. It's our NanoSembler commercial formulation system. You can ask me more about this, maybe at the next break or in front of our booth. It's fully automated. We've basically taken questions, requests from the industry. What do you need? This is a ready-to-use. It's a single-use fluid path. It's automated. We are looking at getting the CFR 21 Part 11 compliance. We're in the process of doing that. It's suitable for use in hazardous locations. Obviously, we use ethanol to make these lipid nanoparticles, so we want to ensure that our equipment is safe. And it provides basic electronic batch records, in-process monitoring. It's quite an impressive system. It's actually about my height, a bit wider than me, and it's about 2 feet deep. It probably weighs about 800 pounds. So it's not something you're going to be able to lift up, but it's on wheels, so you better wheel it in and out of your clean room. So we've seen across all of our scales, and my apologies, it's a bit small, the type is, but we see consistent critical quality attributes. This is after downstream processing of LNPs made at different scales, made on our benchtop instrument, which would be 20 or 30 milliliters, all the way up to batches made in our commercial formulation system, which are in excess of 20, 30, 100 liters or more. So we get consistent particle size, polydispersity, encapsulation efficiency, we also see very similar in vitro and in vivo performance of the formulation. This particular formulation was a self-amplifying RNA-LNP coding for the SARS-CoV-2. And we see this is a mouse model. We see a 42-day prime and boost dose study. We see it across the scales with different instruments, the same performance. Robust immune response is noted for all samples, and consistency between all NanoSembler systems. So that's really important for developing your programs. So you can see here that we are now P&I was acquired about 2 years ago by Danaher Life Sciences. We are now actually part of Cytiva, and together we can provide an end-to-end solution to enable drug development from discovery all the way to commercialization. And you can sort of see where P&I fits in with our novel lipids and delivery solutions, with our API capabilities, working with our partner company, Aldevron, particle formation and downstream processing, all the way to fill finish. And we enable both biopharma companies and CDMOs. There's no technology access fees or royalties associated with any of our instruments. So as you know, developing genomic medicines is challenging. I think together we are tackling these challenges. The knowledge gap is shortening. But again, bottom line is for nucleic acid delivery, especially of large nucleic acids, you need delivery. And that's been said many times over the last 2 days. And we have different needs for different programs. One size does not fit all. So you really have to understand what you're trying to do. So this is a high-level timeline for mRNA-LNP drug development, from basically picking your formulation all the way through to phase 1 clinical trial. It's very important to start with the end in mind. If you're developing an mRNA-LNP drug, you really need to figure out as quickly as possible what your target product profile looks like. Some of those questions are fundamental. Some of them seem more mundane, but they're very important. What's your vial fill? What's the concentration of your drug? How are you going to store it? What's your final buffer? All those questions need to be understood. And you can see here, high level, you can see from program kickoff, working with a client, we lock the formulation, we have analytical process development activities ongoing. Analytics is so important, and that helps you do process development. You cannot do process development without analytics. The other important thing is eventually you're going to need to tech transfer your process to a clean room, most likely at a CDMO. You need that full tech package. Not only the batch record, you need to provide a bill of materials, you need to provide analytical methods and SOPs for operating your equipment. It's a large undertaking, and that has to be carefully planned out. If it's not, it's going to create delays in drug development. Stability is also really important. It's on this slide. If you make an LNP batch, I always recommend put some on stability. It's so important. It doesn't matter what scale. It gives you an early view in the stability of both your carrier system and also your payload. Other things here, having a biological assay, an in vitro assay, especially for mRNA-LNP, is also really, really important to get that established. So back to analytics. mRNA-LNPs are analytically complex drugs. That goes without saying. Whether it's looking at the composition of the LNP, your RNA quality, your raw materials, the quality of the input materials, all very, very important. So I'm just going to call out 2 methods here. Lipid identity and quantitation. It's very important to have a robust method to identify, in this case, 4 lipid components. You have to monitor lipid ratios throughout your process development, throughout your process, that will identify problematic steps. You've also got to look at changes in the lipid ratios. That might indicate compositional issues, where you're losing one of your lipid components throughout your manufacturing process, which is a bad thing. And then also, you might see losses in lipid recovery, and that's telling you you might want to change some of the filters or the TFF cartridges in your downstream processing. So that all needs to be kept in mind. Again, a quick word about the CQA for RNA integrity. We are using a CE-based approach. It is very important, and it's giving us predictable values about there's a direct relationship between RNA integrity and potency. And we have some slides there. We've moved away from agarose gel electrophoresis and other techniques. We're heavily relying upon CE for that. And you can see here how in vitro potency, having that in vitro assay aids in the development of potent formulations. It ensures scale-up processes yield reproducible drug product, and it highlights unforeseen problems that you won't get in other assays, like particle size or encapsulation. So key points, and I'm not going to go through them all, but the punchline is performing 2 days of careful analytics will save you potentially weeks of formulation development time. You need to have reliable analytical assays to generate results you can trust and move forward with. This is my last slide. I just want to show you that this is kind of the journey from preclinical and clinical development all the way to basically tech transfer to a CDMO, making engineering and GMP batches. it's a long journey, and working with us and working with other CDMOs, they can really help you develop drugs effectively. I want to acknowledge over the many years we have received support from the Government of Canada. We're very grateful for that. And working basically with every department at P&I. It's a team effort. Wonderful team to work with. I've really enjoyed the last 5 years I've worked here. So thank you very much for listening. And I don't know if there's time for questions or we wait for the panel, but thank you for your time.
Thank you very much, Lloyd.